Bom dia a todo mundo. É um enorme prazer receber aqui hoje o Jean-Philippe Gibert. Jean é um grande amigo e eu tive a oportunidade de trabalhar com ele no início da carreira dele, né? O Jean, ele estudou na graduação na Universidade de La República, no Uruguai. Aí ele veio ao Brasil, a gente trabalhou primeiro com um estágio, depois isso virou um início de um mestrado, e também pela Universidade de La República, e aí ele teve uma oportunidade incrível de ir para um doutorado na Universidade de Nebraska, Lincoln. E foi um daqueles momentos super interessantes, porque o projeto estava muito legal, mas aquilo ali era uma oportunidade fantástica. E aí ele foi fazer o doutorado, terminou o trabalho do mestrado, né? O artigo foi um artigo que meio que fundou, na verdade, várias coisas no meu laboratório sobre difusão em redes e coevolução. E eu sou eternamente grato ao Gia por ter puxado esse trabalho. Foi realmente um grande desafio e no final ficou super bonito. Ele terminou o doutorado na Universidade de Nebraska. Lincoln foi trabalhar com Justin Ickel, em Merced, na Califórnia. E desde 2017 ele se mudou para a Universidade de Duke e hoje ele é professor lá na Universidade de Duke. E uma coisa que eu sempre fiquei bastante impressionado com o trabalho do Jean, uma coisa que me fascina no trabalho dele, é como ele ataca questões que são não questões necessariamente novas, mas de uma forma muito nova. Então ele traz uma visão sempre criativa, e eu diria até explosiva para algumas ideias, algumas perguntas que se mantêm na ecologia durante muito tempo. Junto a isso, ele também estende a teoria em novas direções. Então, a combinação, acho que do ponto de vista de compreensão teórica, é uma combinação muito única e poderosa, né? Você olhar para novas questões e, ao mesmo tempo, olhar para questões muito antigas ou questões centrais, questões difíceis de resolver, usando abordagens e formas de pensar novas. Então, eu já falei demais. Eu não tenho nem como te agradecer e te expressar o quão emocionado eu fico em ter a oportunidade de ter você de volta no meu laboratório, mesmo que seja virtual. E junto, assim, nesse momento, na minha tela, estão, além de você, a Kate, o Ernesto e o Lucas Medeiros. Então, assim, eu sou um saudosista por natureza, é um pouco do sangue português. Já a sala é sua.
Obrigadão, Mido. Sou eu quem agradece, né? Para mim é um grandíssimo prazer voltar na USP 10 anos depois, né? Faz muito tempo.
Não fica repetindo isso, Jean, por favor.
Mas
Foi
estava
ontem,
fazendo
foi ontem.
contas ontem. Sim, realmente para mim é muito difícil me comunicar em português depois de tantos anos. Eu tento treinar o meu português, mas ele não é tão bom assim. Então a palestra eu vou dar em inglês, se vocês
sem problemas.
Mas realmente eu aprendi a fazer ciência com o Mildo, né? Então realmente sou eu quem agradece tudo aquilo que você deu para minha carreira, é voltar aqui, é ver as caras que eu conheço. Eu leio aquilo que vocês fazem. Eu não conheço alguns de vocês, mas conheço vocês pelo seu trabalho. Então é sempre um grandíssimo honor de estar aqui na USP. So let's change languages. If I'm speaking too quickly, if there's anything that you guys can follow or whatever, feel free to stop me and ask me questions. If for whatever reasons there's any questions you want to ask that we don't get to, feel free to send me an email and I'll be more than happy to email back. And ask questions in whatever language you prefer. I don't necessarily promise that I will answer them in Portuguese, but I will certainly try. So one thing I like to do when I give talks is to talk about stuff that we haven't published yet, right? For many reasons. You know, publishing kind of lags behind science. And so if I were to talk about my published work, it's work I've done 2, 3, 4, 5 years ago. And so it wouldn't necessarily give you an idea as to where it is that we're at now. And that's, I think, what I find to be the most useful for everybody, because it also allows for people to make connections, potential collaborators to see where you're at and where they could make contributions, potential students to see, you know, where they could fit, etc. Right? And so that's what I typically do. So I'm going to tell you 3 stories that are interconnected. These are papers that we haven't published yet. The rest of my papers you can read on my website. They're freely available. They're kind of at different stages of completion. I don't know necessarily that we're going to get to story number 3, but the first story is in revision, the second one is in review, and the third one hasn't been submitted yet. Perhaps the last reason as to why I talk about stuff that we haven't published is, you know, to hear you guys' reactions and feedback. So my job today is to convince you that microbial traits kind of influence food web processes in a changing world. And this is something that I started doing not that long ago, right? And because, as Paulo said, I'm a theoretician, and I found in microbes kind of a window into mathematical tractability and systems that are as complex as any, right? And perhaps just to get something out of my chest, when Paulo announced my talk on Twitter, somebody asked, like, well, is this going to be about actual food webs as well? And perhaps I'm butchering the message. I don't quite remember what the wording was, but the gist of it was that there was this kind of distinction between microbial food webs and other food webs, right? And that's something I just want to say, I don't think makes a lot of sense, right? Because you know, food webs are food webs, and in the definition of those food webs, we don't specify that species need to be, you know, of any kind. They don't have to be metazoans, and they don't have to be microbes, right? As far as you have these networks of species and their feeding interactions, and they account for the flow of energy and matter within those systems, we're talking about a food web. And as a matter of fact, microbial food webs in particular are a component of any food web in the world, right? Just pick one. And with probability 1, it's going to be composed by microbes, period, right? Okay, so now that we're all on the same page, I would say that our story here starts in the boreal peatlands, right? So these landscapes are dominated by these mosses of the genus Sphagnum. These mosses grow very slowly, but they also decompose very slowly because temperatures are so low. And because of that, these boreal peatlands sequester, you know, 35% of all carbon sequestered on in terrestrial ecosystems. And that's important because, as you know, climate is changing, and one of the reasons why that's happening is because of greenhouse gases like carbon dioxide. And so these peatlands actually provide the service, in a sense, they buffer against dramatic changes in CO2. What we don't necessarily know is whether as temperatures rise, they're going to keep on sequestering carbon, or they're going to become, carbon net positive. And they also harbor these incredibly diverse microbial communities. And these microbes play a super, super important but largely overlooked role in this process of carbon storage. And so let me quickly tell you how that works. I have something right here in the middle of the screen. I'm trying to get rid of it. So this is what sphagnum looks like. Uh-oh, 1 second, my pen isn't working. So it's a funny thing, I've been teaching for 1.5 years through Zoom, and I still can't get it to work. So this is what sphagnum looks like. I remember when I was finishing my PhD, I was obsessed with the idea of like studying these moss-associated food webs, because I thought, like, I need an ecosystem in a jar that I can do math with. And I was like, where can I find one? And turns out moss is exactly what I was looking for. And so these moss are inhabited. What's going on? It's like every time I move, I change slides, I have to tell the thing that I'm using a stylus. So these mosses are inhabited by these bacteria that fix nitrogen, and this nitrogen is made available to the moss. The moss takes it up and grows, right? And these are otherwise oligotrophic systems, so there isn't a lot of nutrients to go about to begin with. So moss has an almost, and I know this might be a bit of an offense to Paulo to use this word like this, but an almost symbiotic relationship with moss, with the microbes. And as moss grows, it sequesters carbon. But of course, these same organisms that allow moss to grow, they respire a lot of those that carbon, and all of these processes are temperature dependent. So the balance of whether carbon is going to be sequestered or not depends on whether growth by the moss and low decomposition or respiration by these prokaryotes is stronger. What we don't know is as we crank up temperature, what's going to happen. And I got a chunk of money from the federal government to study actually how, again, my stylus isn't working, it sucks. So how these unicellular organisms known as protists, they don't make phylogenetic sense. They're just a mishmash of a number of organisms that are quite different, but many of these are specialist bacterivores. And so notice that the arrows are pointing in a direction that isn't what, you know, food web ecologists would make it point in the direction at. But I was trying to convince the government to give me this money, so you know, I had to just change the arrows because they were very confused as to why the arrows were pointing in the wrong direction. But the protists actually eat these prokaryotes, and on top of that, there's viral infections both on the protists and on the prokaryotes, and all of these things are temperature dependent. So what we're really trying to understand is, you know, how does this work? How does, as temperature increases, how does these two sources of top-down control on these prokaryotes that are so central to carbon sequestration in these systems, how does that mediate the temperature response of these ecosystems? And so the story today will focus on these predator-prey interactions by protists on these prokaryotes and on the food web processes that that depend on these predator-prey interactions. And we're doing this work every time I need to tell my pen that it has to work. So I guess that I'm gonna stop saying that. We're doing this work in this just crazy site. This is Oak Ridge National Lab. So this is a federally funded lab. And they have this experimental site in the Marcel Experimental Forest up in northern Minnesota, where they're doing something that, you know, only Americans can do. Right, which is nuts. They're literally warming entire bits of the forest in the way I'm showing you here, right? So they have these enclosures, you know, some of them are controls, so they're not doing much. Some of them they're heating at a rate of roughly +2 degrees C, there's +4, +7, +9. You know, temperatures are a bit crazy also. I mean, the predictions right now are, you know, +1.5 to 3 range in the next 50 years. But DOE decided to go way beyond that because at that time, they were suspecting that we could, temperatures could get even crazier than we currently think they are. And so we're literally going into these places and collecting, you know, samples and then studying these little microbial food webs in this experimental site. And this is what they look like on the inside. You know, they have this effectively this spruce trees, and the understory is a combination of a number of short plants, including sphagnum mosses. And they're measuring everything you can think of, right? This is kind of the dream of an ecosystem ecologist, which I'm not, but I'll be okay just studying the microbes that inhabit these sites. But of course, the pandemic. All right, so I'm dripping. I was telling you about this project. I think we're gonna have to cut the talk short, but that's okay.
Jean, just a second. Take your time, okay?
Oh, it's good.
Don't worry, okay?
You know, I do this just because it's fun. And so just randomly when I'm giving talks, I make the internet go down. So I have to run around the building and then I have to give a talk like all drenched in perspiration. So let's keep going. Let's keep going here. So what I was saying is that Pranav actually started the data collection effort for this particular project. He now got accepted at UNC, so he's a college student. Then we've got Adrian and Alex here, who are Duke undergrads. Z, who's a PhD student, and then Sam and Andrea, who are you know, kind of tech people in my lab, and they're finding you know, whether they want to pursue a PhD or not. So what we did is we went to Spruce, we collected a bunch of these protists, which are unicellular organisms, oftentimes specialist bacterivores, not all of them necessarily, but most of them. And these, what I'm showing you here, the names don't matter. Of course, the only thing that matters here are the colors. So what I'm showing you is a gradient of sizes, right? From the smallest Cyclidium glaucoma, which is roughly 10 to 15 micrometers in length, to the ginormous Tildina magna, who's actually roughly 300 micrometers in length. And so we can ask questions about how these protist communities, which prey on the bacteria, might be responding to temperature. And one way to go about doing that is to assess how their population growth is influenced by temperature. And we call this a temperature performance curve. And so we can do this for all of these things, right? There's a lot of information here. Just follow my lead. On the y-axis, when I'm plotting, and I forget my pen only works when it wants to, on the y-axis, we've got the intrinsic growth rate. So the rate at which an individual produces another individual per unit time. This is an important parameter of Lotka-Volterra models and other sorts of mathematical models. If this number is positive, the population grows. If it's negative, the population doesn't. And so we can assess whether population is growing or not across temperatures. And oftentimes you get this sort of like first increasing, then decreasing type of a response, right? And this is pretty common for endotherm species. We call this a unimodal temperature performance curve. And we can do this for a number of organisms, but at the end of the day, what have we learned, right? Kind of not a lot. We can just do, you know, the record keeping, this is how they respond, and that's kind of it. But we don't have real understanding. The other thing we can do is we can image these cells, right? And so we have a machine for which you can put, you know, a water sample and just image just these cells. And every time we turn on that sucker, it just produces hundreds of thousands, if not millions, of individual raster images like this one. And so once we have this, we can measure the length of the cell, the width, and using that, we can calculate things like the size, the shape, and then using the kind of the optical properties of these cells, we can have an idea as to what are the cellular contents of these cells. So effectively, using this, everything is going really weird today. Effectively, we can quantify the simple phenotypes, the simple traits of these cells. Simple, you know, simple is to the eyes of the observer, right? I mean, when we're talking about cellular contents, it isn't necessarily simple, or body size, it isn't necessarily a simple trait, but they're simple to measure, at least in these particular organisms. But once we have these traits, then we can ask a few questions that I find interesting, right? Because we can quantify these traits really well, as I'm showing you here. Notice that we're spanning 3 orders of magnitude. These are the size distributions for all of these organisms. These are the shape distributions for all of these organisms. These are kind of this measure of cellular contents for all of these organisms. And for each of these distributions, we have in the order of hundreds of thousands, sometimes a million individual cells for which we can quantify these traits. And so at this point, we can ask a couple of interesting questions, or questions that I find interesting. Which protist traits may determine the temperature responses of these species separately, right, at the population level? Can we use, you know, any relationship we might be uncovering to parameterize predictive models, and can we use these models to predict how these communities and ecosystems might be responding to temperature changes. So let's address it first. As I was telling you, we can quantify at least these three simple traits. We can quantify these temperature performance curves, right? So in the y-axis, we still have the intrinsic growth rate. We can do that across temperatures, and then, we can characterize the shape of these functions in a number of ways, right? We can calculate the minimum temperature which the population grows, the maximum temperature which the population grows, the maximum, intrinsic growth rate, the thermal optimum, et cetera, right? A number of these descriptors. And this gives us an idea as to kind of physiologic constraints on these temperature performance curves. But we can also ask whether temperature is also influencing other kind of demographic characteristics of these organisms. For example, the carrying capacity, right? So we can do these growth assays where we start these protist populations at different densities. After a few days, we calculate how much they grew, and we plot them against each other. If you remember from logistic growth, the per capita growth rate or intrinsic growth rate, these two things are the same. Sorry that I have different labels. The intrinsic growth rate actually declines with density, right? That's the telltale sign of intraspecific competition, where the intercept is R and the slope is negative R over K, the carrying capacity. Right, so we can do this across temperatures. And so this is what the data looks like.
Yeah, sorry, do you mind if I have a question?
Please go.
Yeah, so the intrinsic growth rate, how do you estimate it from the experiments? Is it like using the curve you just showed or some different way?
Right, so what we do is, let me quickly show you here. We start these Petri dishes with a known number of individuals. It's going to be, well, that should be n sub I. So an initial number of individuals, we'll let it go for 1 or 2 days. And then we count the number of individuals at the end. And so assuming that at first these organisms are growing exponentially, then we can use the solution of the exponential growth equation. And so R effectively is the logarithm of the ratio between the final population and the initial population over the total number of days elapsed, right? Of course, this breaks down if you're close to carrying capacity, right? So you need to do it really quickly in the first few days. But in the first few days, these things are really going exponentially, right? So it works out fine. Does that work, Lucas?
Yeah, sure. That's perfect. Do you just count them or use some other technique? We
count under the scope. And so, but we're talking about really tractable number of individuals, right? So these cells are fairly large. We typically start with 3 individuals in an entire Petri dish, and then we come later on and we count every single individual, right? So it's extremely tractable. There's almost no error in this. Sometimes, but as we count, we pipette the cells. So very rarely we're counting twice. But of course, these things are replicated anyways, right? Does that address your question?
Yes, thanks.
All righty. So we can quantify STPCs for a bunch of these organisms. In this case, we have some 15. Again, if you look at this, it's just a complete disorder, right? There's no clear patterns that emerge or anything. So I'm not hoping you would be able to see anything in here, right? And these are the growth assays that I was telling you about to quantify the slope, right? So the effect on the carrying capacity, and just as a quick kind of rundown of what we get, right? This is kind of R calculated at 22 and calculated at 25. This is K calculated at 22 and K calculated at 25 for a bunch of these organisms. And of course, if we calculate R using this sort of experiment or these sort of experiments, we should get the same. So if you plot them against one each other, they should line up with on the 1-to-1 line. Right? So everything makes sense. We can correctly quantify these demographic parameters and then also quantify kind of the shape of these temperature responses. And the question is, we quantify the traits of these organisms previous to any experimental work. We didn't keep track of any of these traits later on. They might be changing, they may not, we don't know. What we're asking is these traits as they are prior to any warming, any experimental work, are they predictive of any of either the changes in demographic parameters and or the shape of these TPCs. And so what we find is a number of both interesting and uninteresting patterns, right? So let me describe the uninteresting ones to begin with. You know, size, for example, is negatively related to, you know, effectively the intrinsic growth rate. We've known this for a while, right? So it makes a lot of sense that we recover this. It means that whatever patterns we're finding here, at least we're on solid grounds, right? So that's fine. We only have so many species at the same time, but you know, doing this for each species, it's a lot of work. What we didn't necessarily know is that, for example, size would also be related to some other aspects of this TPC, like the critical minimum temperature here, which suggests that organisms that are larger actually have a warmer minimum critical temperature, meaning that they prefer hotter temperatures to come in, right, from extinction. Then there's a few things that didn't quite make sense in my head, like we didn't find, at least in our data, any sort of correlation between size and shape, but these things seem to be anti-correlated, right? Where shape is kind of recovering some of the same patterns that size is. And so what we did here is we did an analysis of commonality to try to figure out whether, even if they're not correlated, whether they were jointly explaining some of these variants. And what happens is something really interesting, which I don't have time to go into, but effectively size and shape sometimes explain the same parameters of the TPC, of the temperature performance curves, but the variants explain changes from parameter to parameter. So they seem to have really joint but independent effects. And then, you know, there are other patterns that are unique to the shape of these organisms. And this is something, there's I think 2 or 3 papers out showing the ecological effects of shape on protists, and that's it, right? So this is the 3rd time we actually find an effect of shape on ecological processes. And so the temperature range actually increases with the aspect ratio, meaning that as organisms get rounder, actually, the temperature range gets longer. We don't really understand why that's happened. And then, you know, this measure of cellular contents, I wouldn't necessarily bet my life on any one of these particular patterns, but you'll see that it allows us to do something really interesting. So there's a few of these relationships that, you know, are unique to some of these traits, but not others. And these are just some of these relationships. I'm not showing them all. But what I want to say here is a couple of things. You know, for example, what if we take out, you know, blepharisma here, this green dot? Well, this relationship goes away. What I'm going to say, though, is that, yeah, we don't have a ton of data. These are only 15 species, but the x-axis, it doesn't get better estimated, right? I mean, we've got millions of individuals, right, per species. The y-axis, it has the error that it has, but there's no reason to be cherry-picking the data here and to be getting rid of some species, but not others. However, we also accounted for that, right? We did this kind of bootstrapping procedure to assess how robust some of these relationships were to you know, just random subsamples of these data. Somebody's calling me on the phone from Nebraska. I don't even know these people. And there seem to be there. Now, the relationships in and of themselves are not what matters. So let me move to the second bit here. So conclusion 1, pre-warming, some of the traits these organisms have seem to be at least correlated with you know, the temperature responses at the individual specific level, right? The question is, well, if these relationships exist, could we, you know, go to the field, measure traits, which are much easier to measure than entire TPCs, and then use that to predict entire community responses and ecosystem level processes responses to temperature. And so how do we go about doing that? What we did is we used a simple model and differential equations, right? So in case you aren't used to some of these, what these things do is they keep track of the change in the abundance of any given species over time, right? The NDT. And that's a function of a number of things. In this case, it's a function of effectively two things. The first bit here is logistic growth, right? It's controlled by the per capita growth rates and the carrying capacity. And the second bit here is effectively interspecific interactions, right? This can be competitive, they can be mutualistic, they can be predatorial. It just depends on the signs of these alpha parameters. Now, here's the thing. We know our traits are at least correlated with the temperature changes in R and the temperature changes in K, right? So we can go from traits for each of these species, we can go from traits to an entire TPC for both R and K, okay? What we don't have any information on is on these alphas. We don't know how these species are interacting. We don't know whether these interactions are changing in any specific ways with temperature. We don't. And the question is, even if we don't know anything about these species interactions, can we say anything about the community level responses of these things just by accounting for the way traits influence the temperature response of these demographic parameters? Yes or no? So these are kind of what running this model forced by the traits, right? For each differential equation, each species, we're plugging in the values that come from our empirical relationships and trying to predict the temperature responses of each species in the community as a whole, right? Again, we don't have any information on the strength of these competition parameters, so we're varying those to have an idea as to how they influence the dynamics. And as one might potentially expect, there's a range of temperatures for which most of the species coexist when competition is low. Diversity kind of goes down in either direction, or richness really. And we can make a number of predictions, including, we expect richness to be maximal at kind of a medium range of temperatures, not too hot, not too cold. Interestingly here, we expect mean body size to increase with temperature, right? Temperature is on the y-axis here. On the x-axis, you can forget about the y-axis, as a matter of fact. It just matters as you move with temperature, what happens with the color. It gets more yellow. And then using the fact that metabolic rates and hence respiration rates scale with body size, we know the body size for all of these species, we know their abundances, we can predict total respiration rates, which are of the entire community, which is an ecosystem level process. And we are predicting effectively this kind of hump-shaped relationship. So we can do this, right? This is math, this is fun, I love doing this, I can do it all day long. And if it was me, I would be like, yeah, we can predict this stuff. Of course, actual communities might have a saying. So yes, we can use these trade, kind of TPC relationships to parameterize predictive models. The question is, are those any good, right? That's where the money's at.
Can I ask something, Jean?
Yes.
So what happened with a lot of the species in high competition? They were like excluded or something?
They are. Okay. Our model says that they would be excluded. So when competition is very strong, you effectively end up with communities that have roughly 4 or 5 species out of 15. And when competition is weak, interspecific competition is weak, you effectively retain all species. Now, again, Kate, we actually don't know anything as to how these interactions are happening or whether they're changing with temperature in any specific ways, right? So big caveat there. Does that more or less answer your question?
And can I ask, how do you, because then you have to, like you imagined the alphas, right? So you just put like equal for everybody or not?
I'll tell you exactly what we do with the alphas in just a second. So, because that's really important for when we test these models, right? What alphas do we actually choose? We need to choose something, right? Because we don't have any information. So how do we test these models? What we do is something very simple. I think this might actually be the only story I tell you today. Sorry about that, guys. So we can we effectively have these 250 milliliter jars. The jars contain just water and a media that allows for bacteria to grow, and bacteria is what the protists eat. And so what we did is we just put in our 15 species in these jars, in these microcosm communities across a range of temperatures, replicated multiple times. And then we asked, are our model predictions any good? And Kate, this is where I answer your question finally. So the question is, are the models any good? Do they line up with the data in any way? We're not feeding the models, we're actually making quantitative predictions here based on the traits of each species and all the species. So what you have here is temperature in degrees C in the x-axis and species richness in the y-axis. And what we do, so the data points here, that's the actual data from the experiment. They're replicated, but of course, these are discrete values, so sometimes they line up on top of one another. The yellow lines just a spline fit, nothing to die for. The gray lines, those are our model predictions. And so, Kate, to your question, what is that we're doing here? We don't know what alphas are. So we're going from 0 to a bunch, right? We're just varying the alphas, because we don't know what they are. We're assuming all of the species get the same alphas, but we've relaxed those assumptions. We've randomly sampled the alphas. We've said everybody gets the same alphas, but we crank them up. We've tried a bunch of things, and kind of no matter what we do, the ensemble prediction, if you will, of our model lies within these gray lines, right? So the dotted lines here are very strong, kind of large alphas, and the solid gray lines are weak alphas, right? And so, as you can see, a model that just takes into account these very simple traits, size, shape, and these kind of cellular contents, and the relationships we uncovered between those traits and the parameters of the TPC, actually make good, not just qualitative, but quantitative predictions as to how these microcosm communities respond, right? What about average trait values? This is what happens, right? We've got temperature in the x-axis again, mean species mass, which in this case we can quantify empirically and then also theoretically. This is what the data looks like, and our predictions are in gray, right? So the data also suggests that increasing mean body size with temperature. We're not suggesting that the species themselves are getting larger. What we're saying is that there's turnover of species across with temperature, and that leads to a change in the composition that results in communities that have larger average body size. And then can we predict an ecosystem level process like respiration? And this is where I was trying to make a segue into the next story, but I'm going to have to tell you that story the next time around. So can we predict total respiration rates? So two things that are very important here. First thing to notice, y-axis, right? They're different. There are two y-axes. There's the respiration rate as predicted by theory. These are, again, quantitative predictions. And there's the actual measured total community respiration rate in the y and in the left axis. You can notice here that they're off by an entire order of magnitude, right? An entire order of magnitude. So the actual data is an order of magnitude higher in total respiration rate than the prediction of our model, right? And then we could also argue that, there's roughly a 5 degrees C, difference in the peak of the predicted kind of maximum respiration rate in what we observe. So what's going on? Why is our model not doing a great job here? Well, because again, these are models that what they do is they tell us what happens with the protists. What they don't tell us is what happens with the bacteria. The bacteria are incredibly abundant in these jars. They're respiring, and so they're adding a ton of respiration. So when I was looking at these relationships, I admit that as a food web ecologist, I was bummed because I couldn't believe that I could predict these relationships without knowing anything about predator-prey interactions and about species interactions in general. was like, no, wait a second, this cannot be right. I should be happy that my model was doing a great job, but I was actually happiest when the model was not doing a great job, because it means, here's an opportunity for these predator-prey interactions to matter. Of course, the next part of the talk was showing you that that was the case, but I'm not going to be able to get there. But next time around, promise that's going to be the case. So the models are pretty good. I mean, they're pretty good. They're pretty good here. They're pretty good here. And, you know, I mean, qualitatively, I would say if I show this to a physicist, they're going to be pretty excited about it. If you show it to an ecologist, they're going to be like, it kind of sucks. So I'm going to say they're good to the extent that they are, but there's a lot that's missing here. And what's missing is it's the ecological interactions. And so I was hoping to get to that part because I'm pretty excited, but we won't. But let me just move forward all the way to the end of my talk here super quickly, so I can thank the people, many people that have been involved in these projects, but also you all for taking the time to be here with us and bearing with us despite a number of things. So thank you all so much, and I'll take any questions you may have if we have any time.
Thank you so much, Jean. I think that we have time for questions. I'm more worried about your time schedule than my time schedule. And of course, everybody in the room can leave whenever they want to leave. So if you have time for answering some questions, so
I do.
I have one that might sound a little bit physicist. So do you think that interactions in your system are playing the role of a scaling factor in the respiration rate? So at the end of the day, the role of structure and the presence, the simple presence of interactions, what it does is to scale up the behavior that you would expect when you just zoom in to the subset of the organisms you actually measure the traits?
Well, so perhaps I kind of mischaracterized what I should have said, really. What I should have said is two things. I think the difference in the scales, right, are due to the fact that our model is effectively missing 80% of the community in there, right? If it's even that low, I mean, protists they get extremely abundant in these jars, but what gets the most abundant, it's bacteria, and they're respiring. It's just a ton of biomass, right? So I'm not saying the interactions themselves are explaining the change in the quantitative predictions, but I think that a combination of the fact that we're missing the bacteria and the fact that the bacteria respond to temperature differentially in the presence and in the absence of the protists might explain some of the change, at least in the x-axis. And as a matter of fact, in the next bit of the talk, I was going to show you that total respiration rates change across temperatures, sure, but they change differentially when the protists are there and when they're not there. When you're assessing how the bacteria activity changes across temperatures, that response changes in the presence of the predators.
Well, that's fantastic. Thank you.
Yeah, no problem.
More questions? I think Kate has had one very interesting about how do you decide to move in that direction. I will let Kate do the proper question.
No, when you were not in the room, we were discussing that I think it's very interesting for people that have worked so long with like mathematical models to feel the need to have a system, and then the transition to like, how was it? Like, did you study a lot of different systems and then pick this one, or was it, I don't know. I don't know what's the order of things, how they happen for you to start working on something that you've never worked before.
Sometimes I look back and I think, where did I go wrong? because I wanted to work with dinosaurs, right? And here I am studying these unicellular organisms that nobody, cares about. But then when I think about it, I'm like, wait a second, is it that I actually went wrong? And in reality, what happens is that I just took a number of kind of paths that locally, right, I found interesting, and they led to interesting questions, and I just followed them. This is some stuff that we're doing currently, and I'm not talking about the other stuff that we keep on doing, right? 75% of the work in my lab is still theoretical. I still work on that interface between ecological and evolutionary dynamics. As a matter of fact, number 3 here was about some of that stuff. But as a mathematician, I always felt like my modeling was kind of falling short of something, right? When I was having conversations with biologists, they typically came back and say like, oh yeah, so, but where did you take those parameters from? Or what kind of traits are you talking about, right? I mean, you can make a model and assume there's a trait and that trait influences these ecological processes. And every time it was like, what trait is that? I was like, well, I don't know, it could be this or it could be that, but for the stuff that I'm talking about, it doesn't quite matter. And they're like, no, it matters. And I get it, right? It does matter in the end, right? And so it was, I didn't quite realize when it happened, but at some point during my PhD, I was working with somebody that was an ecologist, but had mathematical tools. And I was only doing theory, and I was like, why can I not learn from him, right? Why can I not just use this opportunity to just improve some of the areas in which I'm not as well-rounded? And so I was like, hey, John, what about we do some protist experiments? And he's like, what? You doing protist experiments? You're crazy. And I was like, no, come on, it would be fun. It's like, okay, let's do some. And then I sat under the scope, and I was looking at this, little jar with like this petri dish with water. And it was just this crazy amount of activity going on, and I instantly fell in love. I was like, wow, I love lions and wildebeests, and elephants are cool, but there's just so much life going on in these little ponds, and they're tractable. So I could merge kind of the two things, math and data, in ways that I wouldn't have been able to if I was working with lions and wildebeests. So I didn't have to train a lot, right? I'm not a microbiologist. I don't know a lot about protists beyond what I know, and about the ecologist of them, and the fact that we've used protists as a model system for a long time. And now we're starting to realize that they may play a larger role than we thought, just because they're incredibly abundant by biomass. And that kind of naturally led to this tract that I've been telling you about today, about kind of just understanding the dynamics of these microbial food webs. So I don't know if that answers the question. There's no generalities in my answer. It's just this is the path I took. I don't know necessarily that you have to become just a really well-rounded and knowable, empiricist to do some empirical work. I try to interact with talented empiricists that know the systems better than I do. Like, for example, I didn't talk about these people, but Jenny and Marie, right? They're microbial ecologists. They're microbiologists by training. And when I told them about like adding protists and assessing how the temperature response of these bacterial communities would be affected by this predator-prey interaction, they were like, oh yeah, that's awesome, let's do it. And, I was just like thinking in terms of food webs and mathematical models, and they were thinking in terms of DNA, right? Which is something I would have never been able to do in my life. So you know what you know, Kate, that's my point. You know what you know, and you don't know what you don't know. And when you don't know something, you just find the people that know that and that you're excited working with. So my transition was through collaborating with people that knew what they were talking about. And I was just kind of adding my bits and pieces wherever I could.
Fantastic.
That answered my question. Thank you.
Can I, do we have time for one more?
Oh yeah, and I have another one, another question. And Jean, I will be very Brazilian here. If if you need to take off, just say I need to take off.
No, it's good. It's good.
My limit here is the time I need to get up and pick up my kid in the school.
You know, it's the summer here, so I blocked my morning to dedicate to this. So we're good.
Okay,
I really appreciate that, and there is someone that is jumping in a sec. I know he was teaching, and he was very sad that he could attend to the talk, but I think he's just showing up. But Lucas, go on.
Matias.
So here it is.
nice. Yeah, that was super cool talk, Jean. Matthias. Yeah, that was super cool. I had a question about I found really interesting how like just the temperature and the relationship between the temperature and the growth rate could explain like the richness and all those patterns. I was wondering how that is related to the diversity you get in the intrinsic growth rates. Like my feeling is that you would have a higher richness when the R's are more equal across species, and you get lower richness when one of the species has a super high R and other ones have low. So do you see something like this, or?
You know, to be honest, we haven't explored it enough, right? Well, a few things I can tell you is that while the model does a really good job at predicting kind of these, in a sense, meta patterns, right? Richness, right? It actually does a pretty bad job at predicting which species are going to be there and in what abundances, right? And that's something we want to explore next, right? Because to me, that suggests that we're missing a lot of information. This model doesn't take into account, you know, the different types of coexisting, how do you say that? Of coexistence mechanisms, right? We don't actually deal with equalizing or stabilizing processes in the way that, you know, Chesson does, right? We haven't explored any of those questions yet. But what we do know is that, well, we predict, you know, richness, and we predict average body size, and we predict turnover rates as well, which is something I didn't show. It actually does a really bad job at predicting which species are actually going to be there, which is crazy, right? The abundances are off, and which species are there, it's off. For example, in the low, and going back to what you're saying the ones that have the highest intrinsic growth rates are likely to persist, right? And the model predicts, now that I remember, the model does predict that, and I can bring it up. Let me show you here. Share screens.
Yeah, because since like the interactions you were just sampling at random, I was just wondering how the distribution of R's in the community was creating how the distribution of R's in the community was creating those patterns.
Yeah, you're absolutely right. I kind of forgot about it. But so this is the case where competition is this is the case where competition is the strongest. And this is a case where the species that persist are purple tetrahymena, kind of, I don't know what color that is, glaucoma, and then colpoda. And these also are the smallest species and the species that have the highest R. But what we see in reality is that when competition is very strong, it's not those species that persist. It's some other dudes that are bulkier and that actually have lower intrinsic growth rates. And so,
sorry, John. Oh, go on, please.
And so I think your intuition is right? The model is missing stuff. One of the things the model is missing, and that I think is also missing from modern coexistence theory, is the fact that intrinsic growth rates oftentimes give species an edge at the beginning, but at equilibrium, things might be kind of different from what we expect at first. That might be one of the reasons, and we're getting into a kind of worms here, that might be one of the reasons why modern coexistence theory oftentimes doesn't predict coexistence when it does occur. But anyhow, I don't know if I did a good job at answering your question, Lucas, so feel free to retort.
No, that was very insightful. Yeah, thanks. Just one quick, very quick thing. Could you just give a hint of how you would estimate the alphas? Because you can use the traits, right? Like body mass ratio or something like this.
Yeah, it's a good question. It's one that we pondered, right? It's like, okay, how can we estimate the alphas? And so what people typically do to estimate alphas is they do this kind of pairwise competition experiments where they start two populations at similar densities, or sorry, they actually, in theory, they actually do an entire range of densities, right? Some from high densities to low densities for both species, and they go the entire thing. And they use that to estimate alphas. But that's a ton of work, right? Because the combinatorials are ginormous for 15 species, right? You need all the combinations of two, and then you need a bunch of densities. And you need to do that across temperatures.
Yeah, 15 choose 2 plus all the monocultures to get the R's.
Yeah.
I think that there is one option, and I would just jump in because it's related to my question. You have the final species list at the end of your experiments. Do you think that could be possible to try to find the alpha values or the summed effects, direct and indirect effects among species that are more likely to generate the final species set that you observe? So instead of trying to do a mechanistic approach to the problem and try to derive the competition coefficients from the traits and then fill your model with these parameters, you have the final result, you have the list of the species, and then you ask questions about how the pairwise effects of these species through competition and through direct or indirect effects of competition, they are more likely to occur, the values that are more likely to occur given the final set of species that you observe in your experiments.
So correct me if I'm wrong here, Paulo. What you're saying is, run your community experiments, see who is there or who's dominant, and then calculate the alphas for those ones.
Yeah,
and then use that.
I will not say directly the alpha, because the problem is that if you have the alpha, for me, the big problem in estimating the alpha in this case is that the effect of A on B may cascade to B through C, and then turn, in case of competition, this will flip to a positive effect. So instead of trying to assign this effect to actually to the competition coefficients, you might once you have the combined effects through different pathways that connect one species to the other. So it's Matthias and I, we have been doing this with some different classes of models, that you have like a model, a dynamic model for or trait evolution or coextinction or whatever you want. And at the end, we have an equilibrium, and then you can estimate a matrix that describe essentially the combined effects of direct and indirect pathways. It is really strongly related to the Laplacian matrix that you did a lot of work on it. So this might give some insights about the role of interactions, no matter if direct or not, in shaping the outcome in terms these particular outcomes, the set of species and maybe the abundance distribution of species at the end of your simulations.
Right. Yeah, I think you're probably right. It certainly is not something that we have explored. We did explore at some point, just trying to use the data, assuming that at the end, that whatever abundances we were finding were kind of equilibrium abundances and wouldn't change anymore after roughly 3 or 4 weeks of experimental work, which it's likely true for these systems because they go pretty fast to carrying capacity. And then use our models, feed our models to that data to try to extract the overall alphas, right? Kind of the observed after the fact, which include for both direct, but also in many times indirect effects among one another. And what happened is that, of course, you know, ODE feeding has all sorts of quirks, but when you're trying to feed 15 differential equations and, you know, who knows how many parameters, you're screwed big time. So it didn't quite work, but I agree with you that there's a lot more to try in here, and we're just starting to kind of scratch the surface of what this data has to give.
Hoje tem uma pergunta tem a ver com uma questão mais geral em teias tróficas, de uma forma geral. Você vê essas relações interessantes com tamanho de corpo, né, em teias tróficas macroscópicas, vamos chamar assim, e você mostrou algumas relações interessantes, né, com forma e tamanho, né, no seu sistema. A minha dúvida tamanho e forma são as características que governam esses sistemas, ou elas estão correlacionadas com outros fatores que nós não estamos olhando? E me chama a atenção esse ponto, em particular, tanto para interações macroscópicas como microscópicas. Eu penso que nós que trabalhamos com redes ecológicas ainda não usamos de todo o arsenal disponível para extrair efeitos que podem confundir, como, por exemplo, relações filogenéticas, nem tamanho de corpo em grandes organismos está estruturado na filogenia. A mesma coisa, eu imagino, deva acontecer no seu caso. Talvez criaturas com tamanho parecido ou forma parecida sejam mais próximas. E aí, no fim das contas, você mistura o body mass, ele na verdade está carregando junto com ele uma série de outras informações biológicas que não necessariamente tem a ver com o tamanho de corpo, não é mesmo?
Não, eu acho que você tem razão, né? E ainda mais, não só é muito possível para o tamanho do corpo muitas vezes não ser o caráter que está fazendo o efeito diretamente, mas também o tamanho do corpo responde em tempo real às condições bióticas e abióticas. A história número 3, que a gente não chegou, eu vou mostrar rapidinho, só para...
Eu vou ter que te chamar para dar uma palestra só sobre a história número 3.
Que eu acho bem legal. Essa é uma coisa que a gente não está nem perto de enviar, né? Mas eu me fiz a seguinte pergunta, né? Do que a gente podemos fazer um segmento de, por exemplo, o tamanho enquanto as dinâmicas ecológicas acontecem. Então, esse tetraímena é um ciliado mais bem pequeno, tem uns 30 a 40 micrômetros de longo. Então, o que a gente fez foi exatamente isso, né? A gente só botou numa placa de Petri, contou e também mediu o tamanho dos bichos ao longo do tempo. Então, você vê, os bichos crescem exponencialmente, chegam à capacidade de carga, e aí ficam. Mas o tamanho dos bichos cresce, depois cresce, mas significativamente, né? Então, aqui a gente, em promédio, está em, sei lá, 17 micrômetros cúbicos de volume, e no final estamos em 7. Então, mais que 50% de mudança em tamanho meio. Isso vai ter todo tipo de consequências para a forma em que esse bicho interage com outras espécies, mas também tanto comem espécies, mas também são comidos. Então, uma coisa que eu vou dizer é que você tem razão, sem dúvidas, é muito possível que tenha um sinal filogenético forte. Tem trabalhos, tem o trabalho da Anna Eklof, faz, sei lá, faz como tipo 12 anos já desse trabalho, né? Que era Proceedings B ou uma coisa assim, fazendo essa pergunta, tem um sinal filogenético nas redes tróficas? A sua resposta é tipo, não tem, mas tem. Não tem ao nível da espécie, mas tem ao nível de classe, filho, né? Plantas, algas está na base, né? No topo você tem mamífero e ave, e no meio tem artrópodo, né? É basicamente a resposta. Então, sem dúvidas que tem um sinal filogenético muito forte na forma em que o tamanho distribui ao longo dos níveis tróficos. Mas uma coisa que eu vou falar aqui, esse sistema aqui é um classiquíssimo gap limited system, né? Porque eles têm uma boquinha aí, e eles pegam bactéria pela boca, né? Que é chamado de oral groove. Então, se a bactéria é muito grande ou faz biofilm, não entra. Se a bactéria é tamanho certo, aí pode ser comida. E não só isso, tem esse bicho aqui, rapidinho, eu vou mostrar aqui. É engraçado porque eu falo esses bichos como se eu gostara deles, né? Acho que tá me acontecendo ao longo do tempo, mas bom, não, again, não é dinossauro, mas tem esse bicho aqui, é o Plotis. É um bicho bastante grande. Ele tem um oral groove bem grande. Esse bicho come tetraína. A gente sabe isso. A gente já viu isso acontecendo. Se você bota o Plotis numa população de tetraína, eles destruem. Mas eu também sei, porque eu vi no scope, que se o tetraína é muito grande, ele rebota no oral groove. Ele vai, pum, vai para fora. E se o tetraína é pequeno, ele entra e aí, sei lá por quê, pode escapar depois de entrar no oral groove. E aparentemente, não sei muito bem porquê, a gente não estudou, eu não encontrei literatura, mas assim, de forma muito anedótica, o tetraína não pode ser nem muito pequeno nem muito grande. Então, tem uma como seleção por tamanho muito forte aqui. Então, sem dúvidas, em redes tróficas tem uma cena filogenética forte, pelo menos em grandes kind of, níveis, né? Top predators, basal producers, decomposers, coisas no meio. Mas quando você tem interações que são gap limited, aí o tamanho tem uma importância para mediar as interações em tempo real importantes. Mas, como eu estava falando, quando você tem os bichos que estão mudando de tamanho a essa velocidade aqui, então tem muito para entender, né? Muito para entender.
Obrigado, Jean. E assim, só finalizando, eu acho que nesse caso é bem porque assim, falar que também tem uma estrutura filogenética também não responde muito, né? É uma coisa que eu sempre brinco com as pessoas, né? Ninguém interage com ninguém por causa do pedigree, né? Numa teia trófica, né? Não é assim, o fato de você ser, isso acontece porque é um felídeo. Não é esse o mecanismo, né? Na savana, não é porque você é o felídeo que você preda, né? É porque você tem uma série de traços que permitem, né? Uma série de características que permitem que você prede, que você preda. Então, tem esse... me parece que não é um problema trivial separar o que é correlação, que a filogenia acaba trazendo junto, não é isso? No fim das contas, é a não dependência do traço que você estuda, que está estruturada por parentesco, com outros traços. Como separar isso? Eu acho que você mostrou bem o caminho, né? É um conhecimento funcional da biologia, né? O que antigamente o pessoal chamava de biologia funcional, não o que atualmente a gente fala, mas antigamente é a história natural do sistema, né? Então você olha lá, é uma questão de como é a boquinha e a presa, né? Esse tipo de coisa.
Esse tipo de coisa. É uma coisa, eu acho, que chama aí, o Matias Delling e o Pedro têm publicado esse artigo muito interessante, mostrando como o caráter, não me lembro bem dos detalhes, mas como os caracteres explicam a estrutura das redes, tem que ver mesmo com a probóscise, tamanho da probóscise, tamanho você sabe muito melhor do que eu, essa história toda. Mas uma coisa que eu vou dizer, em sistemas microbianos, eu também não mostrei isso, mas tem um monte de informação aqui, tá bem? Então, só dizer um par de coisinhas. Uma coisa que a gente vê é um efeito importante do tamanho do bicho, né? O bicho grande tem um efeito mais importante nas comunidades bacterianas que bicho pequeno. Então, esse Copirium, que é um bicho grande, mais ou menos eles têm essa diferença de tamanho na realidade. Essa é uma bactéria que não tem a mesma diferença de tamanho, é muito mais pequena, na verdade. Mas com esses métodos que os microbiólogos, ecólogos, como se fala isso em português? Microbial ecologists.
Ecólogo microbiano.
Nem tem, acho, essa palavra aí.
Não, nem tem, ok. Essas são coisas que
são biólogos muito pequenos.
Muito pequenos.
São microbiólogos.
Microbiologistas.
Então, isso tudo aqui, eu não tenho nem ideia de como se faz, né? Kate, falando da sua pergunta, eu não faço ideia de como alguém pode fazer esse negócio que eu estou mostrando aqui. Esse foi todo o trabalho dessas meninas aqui, que são as gênias, a Jenny e a Marie. Mas eu perguntei para elas, então, a gente tem... pode calcular o tamanho dos bichos nesses microcosmos. A gente pode mesmo saber se entre tratamentos tem diferença de tamanho. Tem alguma forma de saber se as comunidades bacterianas e os diferentes taxa bacterianos estão respondendo diferencialmente ao tamanho dos bichos? Falaram, sim, tem esse método aí, chama Titan. Talvez o melhor nome que um método pode ter, que não involucra dinossauro, né? Eu falei, a gente precisa desse método, nem importa se é bom resultado, a gente bota no paper, porque eu preciso ter Titã escrito no paper. Mas o que esse negócio faz é, acontece a seguinte pergunta. Se o tamanho do bicho é mais grande, tem alguns clados que preferencialmente vai estar associados com tratamentos onde o tamanho do bicho seja mais grande, ou preferencialmente não associados com tratamentos onde o tamanho do bicho seja mais grande. Então, você pode dizer, bom, esse taxa aí é um taxa que está respondendo à abundância, negativamente, à abundância de Copirium. Mas esse taxa daí, que é um taxa mais, color mais forte, está respondendo positivamente à abundância de Copirium. Esse taxa daí é um taxa que está respondendo positivamente ao tamanho do Copirium. Esse daí é um taxa que está respondendo negativamente ao tamanho. E depois, como você pode fazer uma filogenia, pode fazer perguntas. Então, tem efeitos do tamanho do bicho que preferencialmente afetam positivamente ou negativamente, ou afetam só alguns taxas, mas não outros. Então, no nosso caso, por exemplo, você tem a tetraímena aqui, que é o bicho mais pequeno. Por exemplo, o tamanho da tetraímena afeta espécies no clado esse Armatimonadota. Nem sei o que é o clado, mas o que importa realmente é que, por algum motivo que a gente não entende, tem os efeitos que são clado específico, respostas que são claro específico ao tamanho do bicho. né? Eles respondem ao tamanho da tetraímera, mas não respondem ao tamanho de copídio. Então, eu não estou dizendo assim que essa é a solução de tudo. Mas eu acho que nesse sistema tem uma possibilidade de, não vou dizer assim, separar, mas pelo menos entender a relação entre caractere e efeito filogenético.
E uma coisa legal é que está bem distribuída na filogenia, né? Então, assim, você vê grupos bem diferentes,
Bem distribuído.
tendo efeitos distintos. Né? Isso é bem
Bem
interessante.
distribuído. Mas mesmo assim, tem algumas coisas, por exemplo, tem uns grupos como esse daí, eles não respondem à presença de protista, né? Spiroqueta. Por quê? Sei lá, mas spiroqueta é uma bactéria muito grande, né? Eles têm um formato esquisito, eles são bons nadadores, então talvez seja que eles escapam bem. Ou talvez, sei lá, não são digestíveis, né? Tem algumas diferenças, por exemplo, fala aqui, esse daqui é azul, é o Gematimonadota. Eles respondem preferencialmente a Coppidium, mas não a tetraímera. Então, tem realmente diferenças no que os bichos estão comendo, aparentemente, e como estão interagindo com esses bichos, que talvez nem tenha a ver com os caracteres deles, mas com a história filogenética. Tem muito a dizer aqui, mas, assim, a mensagem mais grande é sim, está distribuído na filogenia, mas tem alguns efeitos que são realmente claro específicos. Tem muito a ser falado, mas a gente está realmente começando a entender isso, né?
E a gente, eu espero em breve ver mais sobre essa história enquanto ela se desenvolve. Jean, muito obrigado por ter tirado um tempinho aí para falar com a gente, para contar um pouco o que você está fazendo. E, bom, seguimos em contato e deixa um abração aí para a Marie e se cuida.
Tem uma pergunta no... Não, desculpa.
Opa.
Não, achei que tinha uma pergunta no chat, mas desculpa. Mas muito obrigado pelo convite. Foi um enorme prazer ver as caras que eu não conhecia, mas voltar a ver as caras dos amigos, Kate, Matias, o Ernesto, pessoas que faz muitos anos que a gente não vê, né?
Acho
Então,
que
obrigado.
2011, né? Eu fico pensando.
Bem
No caso, faz 10 anos. Foi 2011.
10 aninhos.
Você foi
10
embora.
aninhos. Ontem vai fazer 10 anos que a gente fez a escola de redes aqui em São Paulo. Eu tinha prometido quando acabou que eu nunca mais ia fazer uma escola de estudos, mas deu uma vontade ontem de montar isso. Vamos! A Paulinha, lembra que deu essa ideia com a Marília há um tempo atrás. Eu acho que a gente poderia fazer isso.
Vamos, eu adoro me enfiar em fria.
Sou campeão disso também.
Pegar coisa que eu não vou conseguir ter tempo para fazer.
Isso, aí a gente chama o Jean para ser o keynote speaker.
Vai lá.
E já não sei.
Nossa, nessa eu vou nem que eu vá cozinhar para as pessoas, porque na outra eu não fui. Eu fui a única do laboratório que não fui.
Eu sei disso.
Você estava entrando no laboratório, né?
Eu não estava ainda no mestrado, mas...
Você estava entrando no laboratório e eu ainda não... Eu ainda não brigava o suficiente por essas coisas.
Nós éramos jovens.
O que eu me lembro é que faz 10 anos, na minha segunda visita, a Kate está falando a mesma coisa. Eu não fui na rede da universidade. 10 anos depois, ela ainda continua.
Em minha defesa, faz 10 anos que eu não falo sobre isso.
Não, é verdade. Eu pensei em fazer escola, aí eu dormi, sonhei e acordei suando frio, lembrando que a Kate não tinha ido. Triste, posição fetal, assim. Foi
Já me desculpa por não ter visto sua palestra, mas muito bom te ver, viu? Vamos tentar conversar antes de 10 anos.
Pode ser uma meta.
A gente precisa mesmo. A gente convidou o Mildo para dar uma palestra, mas eu vou com certeza eu vou estar convidando vários de vocês.
Vai ser um prazer, já estou pensando. Já estou refazendo a palestra, porque afinal, né, não adianta nada você ter uma palestra já pronta, você tem que refazer toda vez que você dá, só para dar mais trabalho, né?
Eu que digo, hoje à tarde vai ser boa. Saindo daqui. Vai ser a tarde, assim, até eu dar minha palestra, eu tô ferrado. Ficar mexendo slide, mudando letra.
Gente, muito
Até
obrigado.
mais, gente. Valeu.
Bom, que bom ver vocês todos.
Até mais, Jean. Obrigadão. Tchau, tchau, gente.
Tchau.